Imagine, just for a second, all the incredible unseen work happening inside your body right now.
Hmm.
Your muscles, they're constantly generating these forces, pushing blood around, moving food, getting rid of waste. It's this, like, intricate mechanical dance that's fundamental to life.
Absolutely. And we've known for a long time these forces are critical for health, but actually measuring them, especially inside tiny living organs, that's always been, well, a huge challenge.
Yeah, how do you get a precise number on what's happening deep inside something narrow and winding without, major surgery or only looking at the big stuff?
Been a real bottleneck.
Exactly. And that's where today's deep dive comes in. We're looking at this really groundbreaking tech. It's non-toxic and, get this, ingestible.
Ingestible, wow.
Yeah, tiny little optical sensors, they're called the microgauges. And they can actually measure these vital intraluminal forces, basically the pressures and movements happening inside hollow organs. Think your gut, blood vessels, that kind of thing.
And the test subject for this amazing new tool, it's the humble C. elegans roundworm.
Seems a bit random, maybe. A worm.
You might think so, but as we'll get into, its biological systems are surprisingly similar to ours in key ways, making it a really powerful model organism for this kind of work.
Okay, interesting. So our mission today,
Mm.
we're going to unpack what these microgauges actually are, how they work their magic, what they've already told us about how this tiny worm eats, which is fascinating in itself, and crucially, why this might be a really big deal for understanding and maybe even treating human diseases down the line.
Sounds good. Let's dive in.
So first off, why are these internal forces so important anyway?
Well, fundamentally, all those hollow neuromuscular organs, your heart, your intestines, bladder, you name it, they rely on forces being generated just right. Often, it's electrical signals and muscles causing contractions and relaxations.
Right, it's all about controlled movement, pushing things along.
Precisely. And when that goes wrong, the force is too weak, too strong, not timed correctly, or just uncoordinated, that's when you see major problems.
That's what you call motility diseases, right?
Exactly. Conditions where the body's internal transport system breaks down. You see it with food getting stuck in the esophagus or bladder control issues after nerve damage, even things like heart rhythm problems, arrhythmias.
It all comes back to those fundamental forces messing up somehow.
It often does. And our grasp of these internal pressure changes, quantitatively speaking, has just been lacking.
Why is that? What were the hurdles?
Well, a big one was simply the lack of tiny, non-invasive sensors that could actually, get into these tight spots in a living organism.
So lab techniques like optical tweezers or atomic force microscopy, they couldn't really work inside a body.
Not really. No, they're powerful for certain things, but not for in vivo measurements deep inside complex tissues.
Okay, and what about clinical tools like manometry?
Manometry uses catheters, which works for larger organs, like maybe the esophagus or rectum, but it's pretty invasive and not suitable for really small, delicate structures.
Right, and any tiny sensors that did exist needed surgery to implant.
Often, yes. Micromachine sensors typically require implantation. And earlier attempts using fluorescent nanoparticles, well, they had their own set of issues too.
Ah, yeah, the source mentioned that. What was the problem with those older fluorescent sensors?
Several things, really. They could only measure very, very small forces, like in the piconewton range, which isn't enough for many biological processes.
Okay.
They also faded really quickly. That's called photobleaching, so you couldn't track forces for very long, maybe just a few minutes.
Not very practical for studying ongoing processes?
Not at all. And maybe the biggest issue, when you excite them with visible light, the surrounding biological tissue lights up too. That's autofluorescence.
Ah, so it creates a lot of background noise. Makes it hard to see the sensor's actual signal.
Exactly. It just swamps the signal. So the bottom line was the field really lacked a good way to measure these relatively large compressive forces, the kind generated by lots of muscle cells working together, especially deep inside these narrow, complex internal body parts.
Okay, so that sets the stage. Knowing all those challenges, how did these researchers actually build a sensor that could work? What are these microgauges?
So these microgauges are basically optical sensors. They're made of special nanoparticles, tiny crystals that emit light embedded inside very small polystyrene microbeads.
Polystyrene, like plastic.
Yeah, a non-toxic biocompatible type. And the light-emitting part uses something called up-converting nanoparticles, or UCMPs.
Upconverting, what's the trick there?
It's pretty neat. These UCMPs have this unique ability. You shine low-energy infrared light on them, and they emit higher-energy visible light.
Infrared in, visible out. Why is that so useful?
Two big reasons. First, infrared light causes almost zero autofluorescence from the biological tissue itself, so no background noise problem.
Ah, clever. Gets around that issue entirely.
Right. And second, infrared light penetrates much deeper into tissues than visible light, so you can actually see the sensor even when it's buried inside an organ.
Okay, that makes sense. And they picked a specific type of UCNP.
They did. They used a core at shell material. It's a specific chemical formula, NaY0, 0.8Y0, 0.18BR, 0.02F4 at NaYF4, because it offered the best balance between being really sensitive to force and also being bright enough to detect easily. And importantly, it undergoes a visible color change with force that you can actually see with a standard camera.
A color change.
Hmm.
So how does that translate into measuring force? How does a light tell you how much squeeze is happening?
It's surprisingly direct. When you apply compressive force to these microgauges, the ratio of the red light they emit compared to the green light they emit changes. And it changes in a perfectly linear way.
Linear, meaning more force equals proportionally more red light compared to green.
Exactly. As the force increases, the overall light emission gets literally redder. And crucially, this response is consistent and doesn't have hysteresis, meaning the reading isn't affected by whether the force is increasing or decreasing or what happened before. It's a reliable optical readout of the mechanical stress.
That's really elegant. And putting these UCNPs inside the polystyrene beads, that was also strategic.
Very much so. It does a couple of key things. First, being in that hydrophobic bead maximizes their brightness when they're in a watery environment, like inside the worm's pharynx.
Okay.
And second, this is critical for the C. elegans experiments, the bead size lets the sensors bypass the worm's built-in filtering mechanism. Normally, the worm ejects tiny particles, anything smaller than about 200 nanometers.
Ah, but these beads are bigger.
Yes, the average size is around 935 nanometers, which cleverly mimics the size of the bacteria the worm normally eats.
So the worm just gobbles them up thinking they're food.
Essentially, yes, it allows the sensors to get inside without being immediately spat back out. Pretty smart packaging.
Very smart. Okay, so we have these clever sensors. Now, why use C. elegans, the roundworm, as the test bed?
Well, despite being simple, C. elegans has some key advantages. Its pharynx, that's its feeding organ, actually has, and the paper says striking similarities to the human heart.
The human heart, how so?
Things like homologous ion channels that regulate the electrical signals, the action potentials that drive muscle contraction. It's obviously much simpler, but the basic mechanisms share common ground. Plus, worms are just easy to grow in the lab. They're transparent, easy to image using fluorescence microscopy. It makes them a great first step for proving a new technology like this.
Okay, so it's a practical and relevant model. How does the worm actually eat with this pharynx?
It uses a process called pharyngeal pumping. It's basically a muscular tube that rhythmically contracts and relaxes.
Like a little pump.
Exactly. When the muscles contract outwards, the tube opens, sucking in fluid that contains bacteria, its food source.
Yeah.
Then when the muscles relax, the tube closes and it traps the larger particles, the bacteria, and in this case, are microgauges at specific points called trap sites. The fluid and any smaller stuff gets squeezed back out.
And the microgauges are the right size to get trapped but not expelled.
Precisely. The paper notes they're too large to be expelled, but not so large that they cannot enter the mouth. And there's another part too, a grinder.
A grinder. Inside the worm.
Yeah, in the back part of the pharynx, the terminal bulb. It's made of 3 specialized cells that basically mash up the trapped bacteria to break down their cell walls. And this grinding action happens pretty much in sync with the main pumping contractions.
Wow, quite the little food processor. Okay, so they have the sensors, they have the worm model. How did they actually prove this all works safely and gives real measurements inside the worm?
Right, critical steps. First, biocompatibility. They observed that the worms just ingest the microgauges along with their normal bacterial food. The sensors then accumulate in specific parts of the pharynx, mainly the anterior isthmus and that terminal bulb with the grinder.
And they checked if it harmed the worms.
They did rigorous tests. For long-term toxicity and reproduction, they counted the total number of offspring worms produced over 4 days.
Then,
The result was clear. Worms fed microgauges had just as many offspring as worms fed only bacteria. The paper says their fecundity was indistinguishable, so no negative impact on reproduction.
that's good. What about the feeding process itself? Did swallowing these beads mess up the pumping?
Good question. They used electrophysiology, basically recording the electrical signals of the muscle contractions to measure the pumping rate.
Like an EKG for the worm's pharynx?
Kind of, yeah. It's
Wow.
called an EPG, electropharyngogram. And again, the average pumping rate was indistinguishable between worms fed just bacteria and worms fed bacteria plus microgauges.
So the conclusion was,
Microgauge ingestion and accumulation do not alter pharyngeal function. Basically, they're safe and don't interfere with the very process they're trying to measure.
excellent, safe and sound. So next up, calibration. How do you know what force corresponds to what color change?
Exactly. They needed to calibrate the mechano-optical response. They used a pretty sophisticated setup combining confocal microscopy with atomic force microscopy, or AFM.
AFM, that lets you apply tiny, precise forces, right?
Yes. They used it to apply forces ranging from about zero point 0.4 up to 15.2 micronewtons. These forces were chosen because they're in the ballpark of the kind of biological forces happening in vivo, and specifically similar to the pressures thought to be needed to actually damage or inactivate bacteria.
Like the pressures used in food processing almost.
Sort of, yeah. The paper mentions stresses used in high-pressure homogenization to kill bacteria are in a similar range, 5,200 megapascals.
Okay, so they applied these known forces, and what did they see in the microgauge light?
They confirmed that key finding. Applying these compressive forces resulted in that linear increase in the ratio of integrated red and green emission peaks. More force, red or light, in a predictable way.
And was it consistent?
Very. They found the sensitivity was mechanically and optically robust and stayed the same even when they cycled the force up and down 3 times. This showed the response wasn't just some temporary thing or due to heating, and it didn't depend on what forces had been applied before. Super reliable.
Okay, so safe, calibrated, now for the main event. Actually measuring the forces inside the living worm as it eats.
This is where it gets really cool. They set up an experiment to measure everything simultaneously. They collected 60-second recordings where they captured the microgauge color changes using imaging and the electrical activity of the pharynx muscles using the EPG all at the same time.
So they could directly link the muscle firing signal to the force being generated.
Exactly, and they saw a clear temporal correlation. They observed how the microgauges physically moved, pushed back during muscle contraction, then moved forward during relaxation.
Right.
And crucially, in the vast majority of pumping events, 169 out of 185, they saw a measurable increase in force, indicated by the reddening light happening within 100 milliseconds of the muscle starting to relax.
So right when the grinder and traps would be squeezing down.
Precisely. It confirms the microgauges are definitely detecting the influence of muscle activity on intraluminal stress. They're literally measuring the worm's bite force pump by pump.
Incredible. Were they able to put a number on that force?
They were. The average peak change in that red to green light ratio was about 8.2%. Using their calibration curve, that translates to an average maximum force increase of 15.7 9.1 micronewtons.
15.7 micronewtons. Is that a lot for a tiny worm?
Well, they put it into context by estimating the contact stress, that's force over area. They calculated an average stress of about 84 megapascals.
84 MPa. And you said the range to kill bacteria was maybe 5,200?
Exactly. So that estimated stress is right in the range needed to achieve significant bacterial inactivation in lab settings. It matches up nicely with what you'd expect the worm's pharynx needs to do its prime function, as the paper says, which is breaking down food and minimizing bacterial colonization.
So the measurements make biological sense. The worm really is delivering a powerful crunch, relatively speaking.
It seems so. The forces measured seem sufficient to subserve the prime function of the nematode pharynx.
Okay, so this is amazing work in the worm model. But what are the broader implications? How does measuring forces inside a worm help us?
Well, this whole platform creates a framework, a new tool for studying neuromuscular organs more generally, including in disease contexts relevant to humans.
How so?
Think about genetic disorders that cause problems with muscle function or nerve control in organs like the gut or heart. Now, you could potentially model aspects of those diseases in worms, maybe using worms with specific genetic mutations that affect those processes.
Ah, so you could use worms with, say, mutations similar to those causing certain human heart rhythm problems.
Potentially, yes, or mutations affecting gut motility. You could then use these microgauges to directly measure how those mutations affect the actual force generation inside the relevant organ.
And compare it to normal worms.
Exactly, and even test potential drug treatments. See if a drug restores normal force generation in the mutant worms. It gives you a direct, quantitative readout of mechanical function.
That sounds much more direct than just observing behavior or survival.
It is. The paper also suggests this could be a quantitative functional replacement for just looking at muscle structure, like measuring muscle mass loss.
You mean for studying things like aging-related muscle loss, sarcopenia.
Right. Or the effects of diet, like caloric restriction, or testing drugs aimed at improving muscle function or health span. Instead of just seeing if a muscle looks bigger or smaller, you could measure its actual mechanical output. It's a contractile capability from the inside. That could really speed things up.
That's a powerful idea. Are the researchers planning further improvement?
Oh yeah, they already have ideas. They mention using even brighter UCNP materials that maybe have a lower baseline red to green ratio.
Why would that help?
Better signal to noise. It means you could potentially use lower power infrared light for excitation, which reduces any risk of heating the sample during long imaging sessions.
Makes sense. Anything else?
They also want to improve the manufacturing, the polymerization methods to get a tighter distribution of sensor sizes and a more consistent amount of UCMPs loaded into each bead. More uniformity means more reliable data.
Okay.
And maybe implement a way to feed the worms continuously while they're being imaged to make the experimental conditions even more consistent.
So refining the tool further, what's the big takeaway here? What does this capability unlock?
The potential, especially with these improvements, is to directly and non-invasively distinguish intraluminal contractile capability between different groups, say healthy versus diseased models or treated versus untreated, and to really understand that interplay between electrical control and mechanical efficacy.
Getting a handle on how the electrical signals translate into actual physical work inside the organ?
Precisely. It's opening a window onto real-time in vivo biomechanics in a way that just wasn't possible before, especially in these small internal structures.
This really has been an incredible deep dive. These tiny, ingestible microgauges. They genuinely feel like a huge leap forward. We're talking about making invisible forces visible, quantifiable.
Turning them into actual data.
Right. It offers this completely new way to look at fundamental biology and disease. I mean, the breakthrough isn't just the sensor itself. It's the ability to non-invasively get these precise readings from deep inside living systems that were basically off limits before. Imagine, diagnosing something like a gut motility issue, not just based on symptoms or maybe an invasive scope, but by actually measuring the push and pull inside your intestines. That feels like a different world.
It really does shift the paradigm, and for you listening, hopefully this deep dive gives you a real sense of where this cutting-edge science is going. It's a fantastic example of how different fields, materials, science, biology, engineering come together.
Absolutely.
And these kinds of breakthroughs, starting in model organisms like the worm, could genuinely pave the way for new diagnostics and treatments for human health down the road, offering insights we just couldn't get before.
Definitely food for thought.
Yeah.
And it leaves you wondering, given the precision here, measuring these tiny rhythmic forces, what other hidden biological rhythms or maybe internal pressures might we soon be able to decode with tools like this? And what new secrets about health, disease, and aging might that unlock?